Hi, I'm Ada Johanne Ellingsrud, and I just started my PhD in applied mathematics in Oslo at Similar Research Laboratory. The glial cells were for long considered passive bystander of neural activity and have therefore not received the same attention as neurons have over the last couple of decades. But recent research efforts indicate that the glial cells have a way more important and active role than originally thought. And particularly, I will be focusing on the astrocytes' role in brain-water homeostasis. And astrocytes are star-shaped glial cells, as you can see on the sketch to the right there. And in their membrane, there's a high concentration of the water channel protein aquaporin 4, which are the blue squares in the astrocyte membrane. And in particular, there's a high concentration of these water channel proteins in the perivascular spaces surrounding the capillaries within the parenchyma. And as these astrocytes have these water channels, they can easily absorb and release water. And water through these channels are driven by osmotic pressure generated by ion concentration gradients. And studies on mice where they have knocked out the aquaporin 4 show that aquaporin 4 in astrocytes play an important role in clearance of metabolic waste and solutes within the parenchyma, volume control of the extracellular space, as well as formation and elimination of edema. But there are still a lot of unanswered questions regarding the astrocytes' role in brain water clearance. For example, how the macroscopic water flow within the extracellular space affects transport of solutes and metabolic waste, and if the osmotic pressure generated by ion concentration gradient could drive flow in the perivascular spaces. And medical doctors are struggling to give insight or to answer these questions with current measurements techniques, such as in vivo measurements and digital imaging. So mathematical and numerical modeling could potentially help gain new insight, but currently there are limited mathematical and numerical models for modeling astrocyte water dynamics. Such models often take the form of a system of ODEs and or PDEs, and we're looking for variables such as ion concentrations, electrical potentials, volume fractions, fluid velocity, and hydrostatic pressure. And most numerics for astrocyte dynamics are simulating only the electrochemical variables such as ion concentration and potentials, and leave out the fluid velocity and hydrostatic pressure, the more mechanical variables. So as a response to the efficiency of such models, we aim to develop new mathematical and numerical models to connect mechanical and electrochemical features across scales, to study macroscopic fluid flow in extracellular space and how this affects transport of solutes and metabolic waste, and extend the framework also to include flow in the perivascular spaces, which are the blue spaces surrounding the blood vessels, and see if osmotic pressure could be a driving force for this flow. Thank you.